Hello, everyone, and welcome to this audio podcast. This audio podcast is part of Calyx Report Edition. This audio podcast is part of the Bostix SandZtorm, and this audio podcast is part of a Squadron Communique, and this audio podcast is part of a Storm Bostix production. In this audio podcast, in this audio podcast, a little bit in around nephrotoxic nephritis or NTN, NTN and glomerulonephritis, GN in around the basis of the role of chemokines. So when glomeruli are damaged by inflammation, growth growth factors growth factors growth factors are filtered into the urine when where they activate renal sort of the renal tubular cells, causing them to secrete chemokines and cytokines. Mutual stimulation between infiltrating leukocytes attracted by chemokines and fibroblasts gives gives rise to glomerulosclerosis, or where there's glomerular scarring and progressive tubular damage and renal fibrosis. Behind this is the activity of chemokines and receptors. One team had described number one an initiation phase during which injury of renal tubular cells induces leukocyte infiltration. Number two, an amplification phase during which infiltrating leukocytes enhance local production of cytokines and chemokines. And number three, a progression phase during which macrophages in the glomerulus stimulate mesangial cells to secrete fibronectin and collagen type 4, and when there are institutional infiltrates. And number four, uh, a terminal phase of glomerulosclerosis, extensive tubular atrophy, and diffuse kidney scarring and shrinkage. The migration of inflammatory cells into extravascular site requires a series of coordinated signals, including the generation of excuse me, including the generation of chemotactic gradient by the cells of extravascular compartment. The nature of the stimulus and the subsequent spectrum of chemotactic factors determine the specific leukocyte population recruited to the inflammatory site. Members of the chemotactic family play a central role in this process by attracting and stimulating specific subsets of leukocytes. Increasing the understanding of the intracellular pathway that regulates chemokine production in human mesangial cells may provide leads to the design of more effective therapies for the prevention and treatment of glomerular inflammation, glomerular inflammation, and animals genetically deficient in chemokines are resistant to disease induction, although results have not been uniform, with an unexpected accentuation of experimental anti-glomerular basement membrane disease seen in murine or laboratory murine models deficient in CCR1, a receptor for rentase and a macrophage inflammatory protein 1. Chemokines are thus identified as important in laboratory murine models and are clearly active in human disease. Intriguingly, one team had generated had generated CXCR3 deficient laboratory murine models to investigate the functional role of CXCR3 in the kidney under pathological conditions. Nephrotoxic nephritis was induced to analyze the molecular mechanisms of T-cell recruitment in glomerulonephritis by CXCR3 and its three ligands IP10, IP10 chemotactic for CXCL10, MIG chemotactic for CXCL9, and ITAC chemotactic for CXCL11, and to identify potential new targets for therapeutic interventions. So this research identified that the chemokine receptor CXCR3 mediates renal T cell recruitment and subsequent tissue injury in nephrotoxic nephritis. The study provided a rationale for considering CXCR3 blockade in T helper cell type 1 mediated renal inflammation as a future or prospective therapeutic option for recipients with glomerulonephritis or GN and GN subsets. That draws this audio podcast to a close. This has been an audio podcast as part of the Calyx Report edition. This audio podcast has been a part of a Bostix SandZtorm, a Squadron Communique, and an audio podcast as part of a Storm Bostix production.